We're going to move on and talk about antibody-mediated immunity. This is also known as humoral immunity, and it involves an antigen and antibody that interact with each other, and then that antibody is going to in turn neutralize, eliminate, and destroy that foreign antigen that the body has been exposed to. B cells are going to primarily be responsible for the release of the antibodies. It's kind of like when you think back to the chicken pox again. You've had the chicken pox or you've had the vaccine. Your body has built up immunity to the chicken pox, so therefore the next time you are exposed, then you'll either have a minor case or you won't have it at all because of the B cells that your body has specifically produced to provide protection against a particular antigen. You can see kind of by this picture on the slide that the cells have receptors, and once those receptors bind with an antigen, then the antibodies will also be bound to the leukocyte, and that is how they interact with each other and how the body knows that it needs to destroy that. The body learns to make specific antibodies that will provide long-lasting immunity and protection against different bacteria, organisms, and toxins. And there are steps. If you look on page 310 in your Iggy book, you will see the 7 steps that are utilized to stimulate antibody-mediated immunity. So when you look at those 7 steps, it's going to basically begin with exposure or invasion, and that's needed because antibody actions occur inside the body, and for a person to make an antibody that will affect the immune system, they have to first be exposed to an antigen. And then there's antigen recognition. During the antigen recognition, that is the body really seeing that antigen is a non-self cell, and the B cells use macrophages and helper T cells to recognize that this antigen is invading the body. Sensitization occurs next, and that's when the B cell recognizes the antigen and is now sensitized to that antigen. So once the B cell becomes sensitized, it can only be sensitized once, and that allows the B cells to respond to any substance that carries that same antigen as the original antigen. So when you think about that, again, think back to the chicken pox, and once you've had exposure to it, you build up immunity to it. So your body has that antigen recognition to release the B cells that have been sensitized to the chicken pox and therefore are able to secrete the antibodies, and that will in turn help get rid of those antigens or the chickenpox virus. So as I said, it is also sometimes called humoral immunity. One thing to remember also about the antibody-mediated immunity, circulating antibodies can be transferred from one person to another to provide that receiving person with immediate immunity, but it's going to be of short duration. Also be cognizant of agglutination and what agglutination means. And basically, agglutination is a clumping action that results from antibody-linked antigens together, forming a large and small immune complex. Agglutination does not directly destroy the antigen, but it kind of starts the rest of the immune system to begin that defensive effect. When you think about lysis, because we did talk about the immune system destroying antigens and kind of cleaning up the immune system. So when lysis occurs, that's the cell membrane destruction. And with that cell membrane destruction, that antibody is bound to the membrane or the antigen, and that actual binding makes holes in the membrane of the antigen. So when those holes are put into the antigen or virus or bacteria, that makes it less destructive and makes it weaker, and then the body basically will initiate the complement activation and those actions trigger the antibodies to remove or destroy what's left of that antigen. The two classes of antibodies that can activate the complement system are IgG and IgM, and we will discuss those immunoglobulins a little bit in class, as well as during the voiceover presentations. Once the immune system has taken over the antibody or the antigen, then what we see is inactivation or neutralization, and then basically getting rid of what is left of that antigen so that the immune system doesn't become ill. With all of this is how we have sustained immunity, because the memory cells remember the diseases and antigens that we've come in contact with, and so we don't get sick. You'll find this photo in your textbook as well, and it talks about kind of displays to you the immunocompetent patient. So with inflammation, that's going to be a process that provides protection, and then the body must learn to generate specific immune responses when it is infected by or exposed to specific organisms. So with antibody-mediated immunity or humoral immunity, there is an antigen-antibody interaction, and with that interaction, it creates an antigen that was no longer functional, so it neutralizes that antigen, and then will eventually destroy and eliminate the what is left of that protein. Antibodies are produced by sensitized B cells, and those B cells mature in the bone marrow. And then it also needs T lymphocytes or T cells, and that the T cells provide immediate action that is directed at killing the specific antigen. The B lymphocytes and T lymphocytes in turn, once they are sensitized, will become memory B and memory T cells, and they will work once there is re-exposure or subsequent exposures. If we're talking about different types of immunity with acquired antibody-mediated immunity, we discussed earlier that innate native immunity is natural immunity. It is the inflammatory process. When you think about adaptive immunity, that is the immunity that a person's body learns to make or can receive, and is an active adaptive response to invasions by microorganisms or foreign proteins. The active immunity, the body takes part in making active antibodies. So with active immunity, that's the body actually taking an active role, so to speak, in the making of antibodies to help with immunity. Natural active immunity is, again, referring back to your chicken pox, so that antigen enters the body without human assistance. So I get exposed to chicken pox because of droplet exposure. And then the body will in turn build up immunity to that, but there is a natural way in which those triggers or antigens enter the body. And then this natural active immunity is the most effective and longest lasting type of immunity that we do have. And so chickenpox is really the best example that I can give you for that. Acquired antibody-mediated immunity, there are 4 types that you need to be aware of. And with artificial active immunity, that is when an injection or a vaccine occurs or an immunization where that antigen is artificially put into the person's body. So those are going to be our immunizations that we receive. It could be as kids or it could be as adults, such as with the flu vaccine. With passive immunity, antibodies are transferred to the person's body after being made in the body of another person or animal. With passive immunity, there is immediate but a short-term response in the immune system. Natural passive immunity is mom to fetus, so it passes via the placenta barrier, and then also with breast milk when the baby gets that colostrum that is high in the immunoglobulins. The last type is artificial passive immunity. And with artificial passive immunity, that's when the person is injected with antibodies. And then those antibodies provide a very rapid, very short-lived immune response. So with artificial passive immunity, the difference between artificial active is that with artificial active, the person is injected with a virus or with a bacteria. With artificial passive immunity, the person is actually injected with antibodies. And so the best example of that would be with like the rabies vaccines. The person is injected with rabies antibodies, not the rabies antigen or not the trigger. And so then those antibodies will act on the organisms to render them useless and destroy them so that the person doesn't get sick that way. So this is just a little picture or pictorial image of immunizations across the lifespan. So it talks about what is occurring in 0 to 2, 4 months, 6 months, 12 to 18 months, 4 to 6 years, 11 to 12 years, and then finally 14 to 16. So it's really important to promote the use of immunizations to prevent not only the patient from getting sick, but also the community, because with that use of immunizations, our kids continue to build up immunity to those antigens. So your tetanus, diphtheria, whooping cough, measles, mumps, rubella, pertussis, all of those different types of communicable diseases are going to help be prevented with the use of immunizations. We will discuss administration of immunizations and the essentials of administering immunizations in class. Now, on the other side of the immunity spectrum, we have cellular mediated immunity, or CMI, also called cellular immunity. And with cellular mediated immunity, that involves white blood cells, and the different types of white blood cells are utilized to render the trigger or antigen inactive. So there are 4 different types of white blood cells that we're going to talk about. So there are helper T's, and those helper T cells secrete lymphokinins, and we discussed earlier that lymphokinins are utilized to enhance the immune system or the immune response. So the helper T's are utilized to increase maturation as well as increasing the effect of the white blood cells. There are also cytotoxic T cells that are just work on the specific antigen and they destroy it. So when you think about cytotoxic, think toxic and recognize that they do in fact just kill that bacteria or antigen. Cytotoxic T's are very good when it comes to fighting off parasites, viruses, and protozoa. The next type of cell are suppressor T's, and when you think about suppression, think about suppressor T's, and the suppressor T's help keep the immune system in check. So the suppressor T's are really good in preventing hypersensitivity, which will inhibit the normal immune response. And then lastly, we have natural killer cells or NK cells, and those are utilized as far as seeking and destroying and eliminating non-self cells. So the natural killer cells are a very good line of defense when we're talking about cancer and the body getting rid of cancer cells or cells that have what's it called? They have mutations, and those mutations will in turn affect the body adversely. So those natural killer cells are good for cancer cells. They are also very good at fighting viruses and virally infected cells. And again, for total immunocompetence, you have to have cellular mediated immunity, you have to have inflammation, and you have to have antibody mediated immunity. So there are those 3 areas that will encompass a healthy immune system. We will pick up when